Everybody, this is Dr. Nadeem and we are with UNIPATH lectures, the Pursue series. As you're aware, all our lectures are available on YouTube. We also have a Telegram group which is helpful for accessing all lecture related information. You can join the group. We have a Google Drive where the PDF of all the lectures are available. These are the disclaimers. And we are in phase 3, which is recorded pathology lectures, and today we have Pursue 15M, which is hematology erythrocytic disorders, and we are streaming from the famous UNIPATH Speciality Lab, Kolkata, and the topic of the day is erythrocyte enzyme disorders. To talk on this, we have Dr. Mandeep Bedi. She is an MBBS from the famous NRS Medical College and Hospital, Kolkata, a DCP from Kasturba Medical College and Hospital, Manipal, and MD Pathology from Kasturba Medical College and Hospitals, Manipal. She is a consultant hematopathologist at the UNIPATH Speciality Lab, Kolkata, has been an ex-assistant professor at KMC Manipal, an ex-lecturer at NIH Kolkata. Her areas of interest is hematology, and she's got multiple publications in international and national journals. I would request Dr. Mandeep Bedi, ma'am, please start your lecture on erythrocyte enzyme disorders. All yours. Thank you so much.
Hello everyone, this is Dr. Mandeep and I will be discussing the erythrocyte enzyme disorders. These disorders will be discussed under the following objectives of introduction to the basic enzymopathies, the limitations of mature RBCs, metabolism of RBCs, features of enzymopathies, classification, pathogenesis, pathophysiology of a few important enzyme deficiencies, their clinical features and diagnostics, with a conclusive summary. The RBC enzyme disorders are also known as the erythroenzymopathies, which refer to a group of genetic disorders that affect the genes encoding the RBC enzymes and hence affecting the normal function of the enzymes within the RBCs, causing hereditary non-spherocytic hemolytic anemias. They affect the cellular metabolism within the RBCs, and the cellular pathways most commonly affected are the glycolysis pathway, the HMP shunt glutathione metabolism, and the nucleotide metabolism. These enzymopathies are associated with normocytic normochromic hemolytic anemias with insignificant morphological changes, in contrast to other cases or causes of hemolytic anemias where you see a definitive morphology on the smear. Diagnosis is based on detection of reduced enzyme activity and molecular characterization of the defects at cellular level. To begin with, there are certain limitations of the RBCs. As they mature, they are anucleate, so they are incapable of any kind of cell division. They are devoid of ribosomes, so they are incapable of protein synthesis. They lack mitochondria, so they are incapable of oxidative phosphorylation. Despite these, the RBCs survive. They have enzyme-mediated metabolic machineries which are necessary to produce the energy, and specialized metabolic pathways to protect against external oxidant stress. In the absence of all these normal functions, however, the RBC survival is shortened. The RBC metabolism accomplishes several metabolic functions with three main purposes: to maintain both the membrane integrity and the sodium and potassium gradient across the cytoplasm and the extracellular plasma. The energy for this is provided by anaerobic glycolysis, and a large amount of metabolic energy is produced and stored in the form of ATP. Next is to keep the heme iron in its reduced form and to protect the hemoglobin from oxidative denaturation, for which we need the NADPH, which will generate the reduced glutathiones. The RBC metabolism is affected by enzyme deficiencies, and that will lead to impaired physiological functions and reduced lifespan. So before beginning with the enzymopathies, let us know about the normal RBC metabolism. The glycolytic pathway is one of the most important metabolic pathways in the RBC functioning. It produces ATP and intermediates such as fructose 6-phosphate, glyceraldehyde 3-phosphate, and 2,3-biphosphoglycerate. The biphosphoglycerate production regulates the binding and release of oxygen by hemoglobin, facilitating oxygen delivery to the tissues. The lactate production, or which is produced at the end of the cycle, the pyruvate is converted to lactate, which serves as an important energy source for other tissues. The pentose phosphate shunt produces your NADPH, which maintains the reducing environment in the RBCs, protecting them from oxidative damage, supporting the regeneration of reduced glutathione. The RBCs require energy to maintain their structural integrity, deformability, and to support ion pumps that regulate cell volume and electrolyte balance. The ATP generated through glycolysis provides the energy needed for all these processes. So we are saying that it all starts with glycolysis. So let us begin in brief with the glycolysis pathway. In the glycolysis pathway, the glucose is converted into glucose 6-phosphate with the help of the hexokinase enzyme. Now, why this hexokinase enzyme is important here? Because it is a very, very important rate-limiting enzyme. And here, the ATP is converted to ADP, and the kinase is helping us get a phosphate molecule from here. So one phosphate is being donated with the help of these kinase enzymes. So glucose is getting converted to glucose 6-phosphate. The glucose 6-phosphate is further converted with the help of the isomerase enzyme into fructose 6-phosphate. This step is reversible. The fructose 6-phosphate is further converted into fructose 1,6-bisphosphate, again with the help of another kinase enzymes. These kinase enzymes help donate its phosphate group, helps the ATP donate its phosphate group. So till here, it's a very, very important reaction because we are using energy here. These are irreversible henceforth, and it controls the rate of glycolysis. Till here, the glycolysis is the energy investment phase, till fructose 1,6-bisphosphate. So till here, the entire process of glycolysis is known as the energy investment phase. Further down, it is till the production of pyruvate, or till the end of the glycolysis cycle, it is known as the energy generation phase. The fructose 1,6-phosphate is split into the GLAP, or the glutaraldehyde 3-phosphate, and the DAP, or the dihydroxyacetone phosphate. These reactions are reversible, but it is only the GLAP which proceeds further down in the glycolysis pathway. The GLAP is further converted into 1,3-bisphosphoglycerate with the help of hydrogenase enzyme. 1,3-bisphosphoglycerate is further converted to 3-phosphoglycerate, which is then converted into 2-phosphoglycerate with the help of mutase. Now, this step is the first energy producing step in glycolysis. This is known as substrate level phosphorylation. 2-phosphoglycerate is further converted into phosphoenol pyruvate, and finally we get pyruvate. So here the enol pyruvate is converting the phosphoglycerate, is removing the phosphoglycerate to find the PEP. The PEP is further converted into pyruvate. So here the pyruvate kinase is also a very, very important enzyme. If there is any deficiency of this pyruvate kinase enzymes, we get no ATPs. So ultimately, we get the amount of ATP that we can get from this reaction is 2 in number. At the same time, we also notice that the 1,3-BPG via another pathway is converted to 2,3-BPG. Here, it is converted to 2,3-BPG. This decreases the affinity of hemoglobin for oxygen, and there is more dissociation of oxygen to the tissues. The next pathway is the hexose monophosphate shunt, or the HMP pathway. It has two phases, the oxidative phase and the non-oxidative phase. The oxidative phase begins with glucose 6-phosphate and ends with ribulose 5-phosphate, producing enough of NADPH. The basic process here is to get NADPH. So glucose 6-phosphate is converted into 6-phosphogluconolactone. The 6-phosphogluconolactone is converted into 6-phosphogluconic acid, and that is converted into ribulose 5-phosphate. Till here, this is the oxidative phase. The major concern over here is getting our NADPH. The next phase is the non-oxidative phase, where the ribulose 5-phosphate is either epimerized into xylose 5-phosphate, or it goes isomerization into ribose 5-phosphate. So these are the two important steps. Now this xylose 5-phosphate is further converted into fructose 6-phosphate and led into the glycolysis pathway, whereas the ribose 5-phosphate is taken up for the production of nucleotides, especially purine and pyrimidine. As we can see here, this was a rate-limiting enzyme, rate-limiting step in the oxidative phase. Once we get the ribulose 5-phosphate, it is either epimerized into xylose 5-phosphate, which is further taken up and converted into fructose 6-phosphate and taken back into the glycolysis pathway, and it is isomerized into ribose 5-phosphate, which is converted into PR and PP, which is important for your pyrimidine and purine synthesis. A few words about the glutathione metabolism. The NADPH, which we saw was produced during the oxidative phase, is used here to reduce the oxidized form. It causes this reductase enzyme causes the oxidized form to be converted into the reduced form of glutathione. This reduced form of glutathione will detoxify or will detox the body against these free radicals. It converts the H2O2 into water with the help of the peroxidase enzymes. That is why NADPH is extremely important, to maintain it in the reduced form, to maintain the glutathione in the reduced form. To summarize, the glycolysis pathway is important for energy production in the form of ATP, to keep the methemoglobin in the reduced form, to produce 2,3-DPG in the cells so that they can bind to the hemoglobin and decrease its affinity for oxygen so that it is easily available for the tissues. The pentose phosphate pathway is more for production of NADPH. The NADPH is important to keep the glutathione in the reduced form so that it prevents the oxidant injuries, and it also provides our nucleotide enzymes for nucleotide pathways. The RBC enzymopathies are presumed when there is persistent normocytic hemolytic anemias, whereby the hemoglobin abnormalities and anticoagulant reactions have all been excluded. There is absence of spherocytes on smear in spite of it being a hemolytic process. The osmotic fragility is within range. Here, the RBC enzymopathies basically cause hereditary non-spherocytic hemolytic anemias. So before we move on to the enzymopathies, there are certain features which we have to keep in mind. The enzymopathies, the severity will depend upon the importance of the enzyme affected, like the rate-limiting enzyme, as we mentioned in both the pathways. It will depend upon the stability of the mutant enzymes, the possibility to compensate the deficiency by overexpression of the isoenzymes, the functional abnormality of the mutant enzyme or its kinetic property. Most enzymopathies are associated with extravascular hemolysis, though some may also be present with intravascular hemolysis as well. They are clinically heterogeneous, ranging from mild to asymptomatic to chronic hemolytic anemias requiring transfusion. No correlation is present between the residual enzyme activity and the clinical severity. The entire process of hemolysis can be exacerbated during infections. The non-hemolytic symptoms are also seen, especially myopathy in PFK deficiency or neuropathy, severe neuropathy in TPI deficiencies. To classify the enzyme disorders, we basically classify them into according to the three pathways: the glycolytic pathway, the HMP shunt and glutathione metabolism pathway, and the abnormalities of the purine and the pyrimidine metabolisms. So the important enzymes under the glycolysis pathways are the pyruvate kinase, the hexokinase, the glucose phosphate isomerase, the phosphofructokinase, aldolase, triisophosphate isomerase, phosphoglycerate kinase. The enzymes under the HMP shunt and glutathione metabolisms include G6PD deficiencies, glutathione reductase, gamma-glutamyl cysteine, glutathione synthetase. Abnormalities of the purine and the pyrimidine metabolisms include enzymes like pyrimidine nucleotidase deficiency, adenylate kinase deficiency, and an excess of adenosine diaminase. The pathogenesis of enzymopathies. The enzymopathies are inherited as autosomal recessive disorders, most of them, barring a few which are X-linked or autosomal dominant. Mutations alter the structure or the function of the enzyme, leading to reduced or absent enzyme activity. Mutations also affect the production or stability of the enzyme and interfere with the synthesis, the transport, and assembly of the enzymes, resulting in decreased levels of these enzymes within the RBCs. Mutations also affect the active site or other critical regions of the enzyme, leading to loss or alteration of catalytic activity. This leads to deficiency in the enzyme's ability to carry out metabolic functions. The deficiency of specific enzymes can disrupt the normal metabolic pathways, leading to accumulation of toxic metabolites, which have detrimental effects on RBC function, their viability, ultimately leading to hemolysis. Severity depends on specific enzymes affected, the extent of the enzyme deficiencies, the genetic factors which are resulting in mild or asymptomatic to severe anemia and hemolysis or other systemic complications like myopathies and neuropathies. Disrupted metabolic functions or pathways cause impairment of glycolysis, resulting in decreased energy production, decreased lifespan of the RBCs, and their premature destruction. The pathophysiologies of different enzymopathies. We're just covering a few important ones. The enzymopathies in the glycolytic pathway. The first is the pyruvate kinase. The pyruvate kinase is encoded by two separate genes, which is the PKM and the PKLR. The PKM has two isoenzymes, PKM1 and PKM2. This pyruvate kinase enzyme is an important enzyme in the glycolysis pathway. It catalyzes the irreversible conversion of PEP to pyruvate. The enzyme activity here in enzymopathies is reduced. The ATP levels are reduced. The 2,3-DPG levels are increased, and there is apoptosis of RBCs. It is autosomal recessive in inheritance and leads to premature destruction of the RBCs with mild anemias to a full-fledged picture of life-threatening anemias, jaundice, etc., requiring exchange transfusions, and it is one of the most common causes of non-spherocytic hemolytic anemias. Next, we come to the phosphoglycerate kinase, which is encoded by a single structural gene. The importance of this enzyme is it is X-linked. It catalyzes the conversion of 1,3-DPG to 3-phosphoglycerate conversion. This is one of the two glycolytic reactions resulting in the net ATP generations from ADP. An alternate fate of 1,3-DPG is the formation of 2,3-DPG, which is an important intermediate that enhances the oxygen release from hemoglobin. Now, this is encoded by a single gene, PGK1, and biochemical variants have all been reported because of missense mutations. This leads to severe hemolysis in males and variable hemolysis in females, at the same time presents with neurological diseases. Hexokinase. Hexokinase is the first enzyme in the glycolytic pathway and catalyzes the conversion of glucose to glucose 6-phosphate. It has a very strategic role in the regulation of glucose consumption. There are 4 hexokinase isoenzymes, of which the hexokinase 1 encodes for the RBC hexokinase. The inheritance again is autosomal recessive. The clinical features, though not well defined or well characterized, varies from fully compensated hemolysis to hyperbilirubinemias to splenomegalis to formation of pigment stones, but there are no unique RBC morphological features. Next enzyme is the glucose phosphate isomerase, or GPI. It catalyzes the conversion of fructose 6-phosphate to glucose 6-phosphate. The GPI pathogenic variants are basically present because of missense mutations and gene deletions. There is a variable severity of hemolysis here, and the it can lead to something like hydrops fetalysis in a newborn or in the fetus, and it also leads to neuromuscular impairment. Again, the inheritance here is autosomal recessive. Next, we come to phosphofructokinase. The phosphofructokinase has 2 subtypes. It has 2 subtypes: the muscle or the M type and the liver type subunits. It catalyzes the phosphorylation of fructose 6-phosphate to fructose 1,6-diphosphate, which is another rate-limiting enzyme in cases of glycolysis. It is a tetrameric protein, and it is made up of a combination of these muscle and the liver subunits. The variability will depend upon the diverse symptom. The variability, because there are variable types, that explains the diverse symptoms which are associated with this deficiency state. So it results with congenital non-spherocytic hemolytic anemias and myopathies, which is the Therese disease. The lack of M subunit leads to unfavorable shift in the oxygen dissociation curve and leads to hemolysis. The next enzyme is the aldolase. The aldolase is important in the conversion of fructose 1,6-diphosphate to dihydroxyacetone glyceraldehyde triphosphate. This is again has an autosomal recessive inheritance. It has various presentations from hemolytic anemia to delayed development to psychosomatic retardations to jaundice, myopathies, transfusion-dependent anemias requiring splenectomy, and also psychomotor retardations. Now, there are 3 isoenzymes present here, of which the aldolase A is important in cases of RBCs. Next, we come to the enzymes in the hexose monophosphate shunt and glutathione metabolism. The G6PD is a very, very important enzyme, and it is the rate-limiting enzyme in the HMP shunt. I will not go into details of the G6PD disease pathology. I will just mention a few words about the enzyme and the symptoms and the diseases it causes and why so. There is reduced activity reduced activity of this enzyme leads to reduced production of NADPH. The ability of RBCs to regenerate the reduced form of glutathione and combat the oxidative stress is hence impaired. So this makes the RBCs more susceptible to reactive oxygen species damage, and this accumulates in the cell. This is one of the conditions which is X-linked. So there is trigger-induced symptoms. There will be hemolytic anemia. There is features of intravascular and extravascular hemolysis in G6PD. And at the same time, we are able to see the Heinz bodies on the peripheral smear. Next, we come to the glutathione reductase. Glutathione reductase is an important enzyme in the reduction of glutathione in the presence of NADPH. The reduced glutathione contains the FAD activity. FAD is the flavin adenine dinucleotide as a prosthetic component, and hence the normal enzyme activity is dependent on the dietary availability of riboflavin. The hemolysis, you get a history of hemolysis post-fava bean ingestion. The inheritance is relatively unclear here. Gamma-glutamyl cysteine synthetase has a light and a heavy subunit. It catalyzes the first step of GSH synthesis, and it basically functions of missense mutations. There is chronic hemolysis, there will be jaundice, splenomegaly, neurological symptoms, and stones. Glutathione synthetase, or GSS, it catalyzes the second step of glutathione synthesis, and it is pathognomonic variants in GSS genes. The features will be the same: hemolysis, metabolic acidosis, and neurological symptoms. The enzyme abnormalities which are present or seen in cases of purine and pyrimidine metabolism, the most important being pyrimidine 5' nucleotidase, which catalyzes the degeneration of cytidine and uridine. The deficient reticulocytes. Now, what happens is the reticulocytes deficient in this particular enzymes accumulate large number of C and U compounds. These C and U compounds, as a consequences of what these impaired ribosomal degranation, they are and they cause intracellular aggregations in the form of P5-N deficient cells and appears as basophilic stipplings in the cells. Cases of pyrimidine 5' nucleotidase deficiencies were formerly called as high ATP syndromes, owing to the erroneous assumptions that the large amount of nucleotidase was adenine phosphate rather than pyrimidine phosphate. Next is the adenine deaminase excess. Here we are not talking about a deficiency, we are talking about an excess of this enzyme. This enzyme catalyzes deamination of adenosine. So adenosine is a common substrate for two enzymes, adenosine kinase and adenosine diaminase. So excess ADA causes deficiency of ADK. So here a pathological variant causes hyperactive ADA and depletes the adenine, leading to hemolytic anemias. In contrast to the ADA excess, ADA deficiency is associated with an autosomal recessive condition of severe combined immunodeficiency. This is an autosomal dominant condition because of the excess. When there is a deficiency of ADA, it leads to an autosomal recessive condition that leads to severe combined immunodeficiency, SCID. The adenylate kinase is an important enzyme that catalyzes the interconversions of adenine nucleotidases and help in salvaging the LMPs. It has 3 isoenzymes, of which AK1 is the isoenzyme in cases of RBCs, and they also cause non-spherocytal hemolytic anemias. The diagnosis of the enzymopathies involves a clinical evaluation, a family history, the episodes of anemia, jaundice, or the presence or absence of hemolysis. In cases of myopathies, neuropathies, they will all be features and clinical symptoms present. So a very thorough, a brief history is extremely important in these cases. The complete blood count and smear for morphology. Most of them do not have a significant change in the peripheral smear. You can see some white cells, you will be able to see some blister cells or half semi-ghost cells, or you'll be able to see Heinz bodies. Now, they are very pathognomonic of certain conditions, like Heinz bodies are seen in glucose 6-phosphate deficiencies, and basophilic stippling is seen in P5' nucleotidase deficiency. The reticulocyte count, again, is an indicator of the degree of hemolysis, and it reflects on the red cell turnover and premature destruction. The enzyme activity assays, the quantitative spectrophotometric analysis helps assess the amount of enzyme that is present. Genetic testing includes the PCR, direct sequencing, denature, gradient, gel electrophoresis for specific mutations that we're talking about, and functional tests measures the metabolite intermediates in these enzyme deficiencies like G6P or PK activities. To conclude, enzymopathies can affect various organ systems and have diverse presentations ranging from mild or asymptomatic anemias to severe and life-threatening ones requiring transfusion therapies. Understanding the pathophysiology of enzymopathies is very crucial for accurate diagnosis, appropriate management, and genetic counseling. The genetic testing and the enzyme activities play an important role in confirming the diagnosis and determining the specific enzyme deficiencies. Management of these enzymopathies involves avoiding all kinds of triggers, be it medications, be it dietary components, or environmental toxins. The enzyme replacement therapy or supportive treatments may be necessary to manage symptoms and prevent the complications. Last but not least, genetic counseling is essential as these conditions all have a hereditary base or nature. Thank you.